one is male, one is female, and they have 4 pups. I don't know which one of them are mutant or wild type or heterozygous. So to know which one is mutant, wild type, and heterozygous, I have to do genotyping. For that, I will cut tail samples from all of the 4 mice, and I will put these 4 tail samples in 4 tubes, put 50 millimolar sodium hydroxide, and the quantity will be 75 microliter. I will heat it at 98 degrees centigrade for 60 minutes, and then I add 75 microliter of 1 mole hydrochloric acid, vortex it, and centrifuge it. DNA is isolated. But I am actually doing genotyping for example, gene A. The quantity of the gene A in this sample is very less. So first, to detect that gene A is either mutated or it is wild type, I need multiple copies of that gene. For this, first I will do PCR. I will amplify my DNA. And for that, I will take 1 microliter of the that DNA samples in these 4 tubes from all the 4 animals, and then I will add there take buffer, magnesium chloride, DNTPs, forward and reverse primers, and a take polymerase, and then I will add some water and I will put these tubes in PCR. I will set the denaturation temperature to 94 degrees centigrade, annealing to 58. It may vary, maybe 60, 62 based on the umbilical size of the gene, and elongation temperature to 72. Usually the 58, this annealing temperature depends on the annealing of the primers. That's why it varies from gene to gene. But the denaturation and elongation temperature are quite same for all type of gene. And then I will set the temperature to 35, set the cycle to 35 cycles. And after 35 cycle, I will get 34 billion copies of that gene of interest, so that I will run it in the electrophoresis, gel electrophoresis, and then after running it at 100 volt for 70 minutes, I will scan the gel in this Lecore imaging system. Now I have these bands. For example, this is heterozygous, this is heterozygous, this is wild type, and this is mutant. Now I have a clear idea that out of these 4 animals, which one is mutated animal, and which animal has mutation and which animal is wild type. So I will compare these two, wild type and mutant animals. So now I have wild type animal and I have mutant animal, and I am comparing both of them. My question is related to a skeletal system. That is why I am taking osteoblast, and I have mutated this gene. So obviously my question will be, if I mutate one gene, what will be its effect on the gene B, C, D, E, and different other genes. For that I need RNA. So I will crush the bone, add some trizol and stainless steel beads, and then I will bullet blender it and add chloroform and spin it so that the supernatant has now RNA. I will put this supernatant into another tube and add isopropanol and refrigerate it at -20 degrees centigrade for 1 day, or even one hour is sufficient. Then I will spin it, and in this way I will isolate RNA. Now this RNA has to be quantified because I have now 2 animals. One is wild type and one is mutant. So I have to take equal quantity of the RNA. For that I will use this nano drop. I will put 1 microliter from each sample here, and it will give me a reading, or it will give me the quantity of the RNA in the form of nanogram to microliter, and I will quantify it so that I will get the total RNA of sample A and then B and C. For gene expression analysis, this RNA is unstable, so first I have to make a cDNA of it, For making complementary DNA, I will put this RNA in 2 tubes, 1 microgram from each sample into 2 tubes, and then I will put some reverse transcriptase there and some buffers there and set the PCR so that the reverse transcription happen here. So after 30-40 minutes, I have complementary DNA, and this complementary DNA I have to use for the gene expression analysis or qPCR. So I will quantify it and I will go to the